you as our speaker. So he's joining us from the University of Hull, works on the science of fisheries management, and describes himself as informal, sometimes irreverent, but really committed to helping us understand what is a very complex system, but something that's incredibly important and fragile. So I'll hand over. Thank you, Magnus.
I think last time I did one of these kinds of presentations was to the Scarborough field gnats, and I've done a few to them. I usually count having 30% of them still awake by the end of it as a victory. So I might break that today. Hopefully you'll enjoy the presentation. I am going to take a meander through some things that I just think are interesting, and some things that I think are worrying, some experiences that I've had, and they're all stitched together into something that will lead to a nice question, a sort of slightly controversial, hopefully, question at the end. So, is that showing the slide? I can't believe that's showing the slide because it's different on here. Never mind. Anyway, if you have a mobile phone and it's online, if you go to menti.com, that would be really helpful, and put in the code number which is there, which is 612244001. And I just want you to answer that question. I just want you to tell me, from personal experience, don't think about the science so much, but from personal experience, what one thing in the environment has changed in your lifetime? What's changed? What we should see when you respond to that are some words coming up here. So you should be asked to put, oh, good. I'm very interested to see what comes up here. So there's some really interesting things on what we generally find when we use this at university for teaching. And what's generally interesting is that things come out and you think, oh, I hadn't thought about that, or I haven't thought about that for a while. And I think the things on here about insects is really interesting. Less insects on windscreens, I think, is something that we can probably all identify with. But the thing that I think you can all agree on is that there is change occurring, and it's occurring within lifetimes. It's occurring in sort of 10-year scales. Fish are moving north in the North Sea at a rate of 30 kilometers every 10 years. And fishermen are starting to experience the difficulties around changing stocks and perhaps legislation not quite keeping up with that. But things are changing. There was a lionfish caught off the south coast of England recently. Lionfish, they're supposed to be in the Indo-Western Pacific. They've invaded the Mediterranean, which is frankly a basket case now, and in the Caribbean. And I think James mentioned the triggerfish. The triggerfish, yeah, and the farns. Yeah, I mean, what's that doing there? Triggerfish in the farns. And then there was a fisherman recently found a Dover sole way up in the North Sea. So things are changing. We're starting to find things in places where perhaps they should. So I'm going to jump around a little bit. So I'm going to go back to my PhD, which I'd started many, many years ago. And I'm going to talk to you about shrimp eyes. And you'll see why it becomes relevant in a while. So the biggest dance in the world of animals is the daily vertical migration of pelagic organisms in the ocean. Okay, so they come up to the surface at night to feed, and then when the sun comes up, they sink down to hide from predators and to digest their food, and then they come back up again. So down and up and down and up. It's a really important part of the global carbon cycle, and the things like the lipid pump and the carbon pumps, sucking stuff from the surface down into depth, and then it gets captured by processes at depth. I did a PhD on shrimps, some like this one. So this is a beautiful animal. I think it's beautiful. This is Narcosis. It's a big animal. It's like this. They're red. They're quite often red because that's like black in the deep sea where there's no light really. So being red in color means you're invisible generally. They're oily. They're quite diffuse. They're spread out. They're not the sort of thing you can fish for because they're really well spread out. And I focused on the eyes of these animals. So I spent 3 years looking at the eyes of deep sea decapods. Okay, if you read my abstract, which is about the reflecting superposition compound eyes of Natantian decapods, you wouldn't. I don't understand it anymore when I look back through it. It's ridiculously jargonesque. But one of the things I was interested in was the design of shrimp eyes. And one of the things that shrimps have is a tapetum. Now, tapetum is something that you find in lots of animals. So here's some examples of crocodile, cat, and a deer. And you can see they've got shiny eyes. Okay, so when you try it tonight, when you go home, grab the cat, but scruff the neck, Shine the torch in its eye, and you'll see this lovely reflection. And you can tell quite a lot from that reflection. And the tapetum has a purpose. Now that's the bit that's at the back of the eye, this mirror at the back of the eye. And it basically, the back of the eye, the retina, has photoreceptors in it, which are cells that are a particular length. Okay? And as light passes through those cells, it gets absorbed. If the cell is that long, a certain amount of light gets absorbed. If you want to absorb more light, you would have to have bigger receptor cells at the back of the eye. That means you'd have to have bigger eyes. Now, there's a sort of balance there between how much light you can absorb and how big your eyes are. Now, if you're a shrimp that's this size, and you don't really want to have eyes at the size of my hands, do you? That's not going to be very good. So if there's a way of minimizing the size of your eyes, that's useful. So what you can do is you can put a mirror at the back of your eye, okay? And that basically doubles the path length that light has to pass through in a photoreceptor. So you can minimize the size of the eyes. You all with me so far? Yes? Okay. If you get lost, stick your hand up. Right. So that's a picture, at the bottom one there is a picture of a photoreceptor with a mirror, which effectively doubles the length of the cell. So it means it doesn't quite double the amount of light absorbed, but it enhances the efficiency of the eye. So cats have it, cows have it, deer have it, frogs have it, crocodiles have it, people have it, and shrimps have it as well. The thing about deep sea shrimp eyes, though, is that the pattern of that tapetum, which enhances the efficiency of the eye, varies. So animals which are found close to the surface only have a tapetum on the underside of the eye because there's sunlight shining down. Okay, sunlight shines down. If you've got a shiny top of the eye, that's not very good if there's predators around because they'll see all these little sparkles in the water. Oh, food, grab it. Okay, animals that are found in deep water, very deep water, have a tapetum that covers the whole of the eye because there's no light there apart from bioluminescence. So they can quite safely have an eye that's very efficient all the way around. And there's quite a nice pattern with depth. So as you go deeper, the amount of tapetum, that reflective surface in the eye, goes from so in the shallow water, it's just on the very bottom. As you go deeper, the tapetum spreads out. And that means you can start to make a judgment about the depth at which a shrimp comes from by how shiny its eyes are. And that's quite useful, because there's not much known about these guys. So we can start to work out what sort of depths animals live at and what the interactions might be with other animals. Now, we were looking at one, the problem with having a shiny eye at depth is that you may encounter something like this. And this is a tolla, which is a jellyfish. It's about this size. You can see it's quite bright. It's bioluminescent. These are sort of major predators in the deep sea. Jellies are a really important part of deep sea ecology. And the bioluminescence that comes off these things is bright enough to read a newspaper by. So here's one when it's been poked. So you imagine you're a shrimp in the deep sea with your big shiny eyes swimming around. You bump into a jellyfish and it does this. You get this bright light. It's really bright. It doesn't show it really. It's bright. You can read a newspaper with it. If you've got shiny eyes, that means there's going to be light coming from the jellyfish and coming out of your eyes. And what that means is you're then vulnerable to predation because you're swimming around with a nice bright shine coming out of your eyes. And we looked at some shrimps and we found that rather than having that sort of change in pattern of reflectivity from the bottom to the top, the eye had a hole in the reflective bit. Okay, so it was like this bit down the bottom left-hand corner, this yellow and black bit. So that's a hole in the tapetum, the hole in the mirror. And we could scratch our heads for a while. Why is that? Why would you have a hole in the middle of your eye? Why would you have a dark patch? If you're a deep sea shrimp and you need to be able to see well all the time, and you can have a tapetum all round usually. Well, what we worked out is that shrimps blink. Okay? And when they blink, it's a way of hiding. So if you look at the diagram there, that's looking down at a shrimp, and you can see the two eyes. One of the eyes is sticking out like this, and the other one is pulled forward. So the shrimps have a bar that runs across between the two eyes, and a muscle that's attached to it. And when you pull on that muscle, the eyes go from there to there. And when you scare a shrimp, you may have seen this, I don't know, telly or something. But when you scare a shrimp, it swims backwards really quickly. It flips its abdomen and runs away. So they close their eyes and they run backwards. Okay? That's my impersonation of the shrimp. And when they do that, if this particular species is called a Acanthophira purpurea, if I remember rightly. So they have that black hole in the middle of the eye. When they do that, and they swim backwards really quickly, they move away from the atolla, and they're hiding. They close their eyes, it's a good way of hiding. I just think that's really cool. And it does become relevant later on, but we published a paper on that, and I'm so proud of that, just to publish a paper on the fact that shrimps blink is amazing. I actually have some black and white footage here. Hopefully, if it'll play. Yeah. So this is looking down on the shrimp. This is on the RSS Discovery, and you can see the eyes moving in. But it's actually been poked by my supervisor with a pair of forceps. Peter Shelton. There you go. It likes it really. It's like a massage. Okay, so this is a view from the top. Now this is a view from the front. So you can see the eyes are shining because there's a light shining into them. And then if you tweak its nose. Da, da, da. Yay! Right? It disappears. Okay? I just think that's amazing. It's such a clever adaptation that these animals have. So I may be alone in that, but anyway. So I was looking at shrimp's eyes, and it sort of led me on thinking, looking at the eye structure is all very good, and we found some cool stuff out and all the rest of it. And then I sort of finished my PhD, and I was looking around for something else to work on. And so I started working with these guys. Anyone know what it is? Anyone identify that? Antarctic dominant species. Yeah, Euphosia superba, the krill. So it basically does the same job as copepods do in the North Atlantic. And what's interesting about this animal is it's behaving, okay? It's doing stuff. It looks very purposeful. The other thing that's interesting about krill is that when they stop swimming, they sink. And that's a bit of a disadvantage if you are a pelagic organism and every time you stop swimming, you sink, right? That means you've got to swim all the time. That means you've got to swim all the time, 24 hours a day. Krill don't rest on the seabed. The other thing is, so you can see it's slightly sinking there. It's also very beautiful. I think they look like galloping horses. This is on YouTube. Catch me after if you want any of the links or data or anything from what I'm presenting today. So I started working with krill, and what I was really interested in was what happens at the level of the individual. Most of the stuff that's understood about krill is taken from, we go out on the Discovery or the Boaty McBoatface, which isn't called that, it's called the David Attenborough, isn't it? Sir David Attenborough. You go out on that and you catch krill in nets at certain depths and you work out, and look at what they've been feeding on, what the density of the swarm is and how big they are and whether there's males and females. But there's not very much known about what does an individual krill do. What do they do through the day? How do they interact with other krill? Because you can't just jump in the water. That's the only footage I've seen of a krill kind of swimming around, and it's quite close to shore, obviously. But you can't just jump in the water in the Antarctic. All sorts of things there might eat you. And it's very, very cold. If there's any divers in the room, if you've ever been in, what would we get, 8 degrees centigrade here, that's painful. Just imagine 2 degrees centigrade, diving in 2 degrees centigrade. And you can't chase a krill, because a krill can swim really fast. So you can't observe. So it's not like bird watching. You can't go out with your binoculars and sort of look at krill and see what they do during the day and analyze their behavior. Oops. So what I did was I put krill, thank you, Lauren. This is Lauren's equivalent of a krill. I put krill on a pendulum, okay? And I was working in Kristineberg, it's a marine lab in Sweden. And krill, they swim. So what this pendulum allowed me to do was to understand what happens at the level of the individual krill. So if a krill's not swimming, I know this is a lobster, okay? So, if a krill's not swimming, it will just sit there. Nothing happens. If it swims a little bit, the pendulum moves, okay? And then if it stops swimming, the pendulum swims back again. If it swims enough to hold its own weight, then it'll be at 90 degrees. If it swims enough to hold its own weight, it'll be at 90 degrees. About half its weight, 45 degrees. Not very much, okay? Right, so krill behavior can be monitored using these pendulums. And so what we did was we looked at how this pendulum moved with a krill on it. And we were able to see all sorts of things. We were able to look at the behavior of the animals over time. We found all sorts of interesting things out. That female krill, when they've just molted, so the krill that's found in Sweden is called Meganictiphines. It's not Euphosia, it's an analogue of the Antarctic species. Females that have just molted sink to the bottom because they can't swim. They've just molted and the skin's all floppy and they just fall down through the water. And the males jump on them straight away, okay? Because they're defenseless. It was interesting, So we got all sorts of things out of this. We were able to look at how fast they beat their pleopods. We were able to look at how their behavior varied over time, how much they swam, when they swam, whether males swam more than females, what the beat rates were, what sort of signals would they be generating that other animals might pick up on. There's a whole story there that I won't go into. And I thought, okay, doing it in Christineberg was fantastic. It's a beautiful place. It's a kind of marine biology mecca in Europe. And I thought, well, I'd quite like to do it at sea. Let's go and measure the behavior of krill on that time would have been the James Clark Ross. This is a picture from the James Clark Ross in the Antarctic. Anyone see a problem? So I might want to measure the behavior of the krill in this kind of slightly. It makes me feel a bit. You want to read what you want to read and follow the book. So I had to think of a clever solution to this problem. I think so. This James Clark Ross unit, 4 stories up the bridge, okay? And at one point, I think it's here, water travels 50-200 feet from the bow of the boat. Really interesting working at sea. You get used to things moving all the time. You get used to putting your coffee cup there instead of here. You get used to holding on to things as you walk around. You get used to knowing everybody that's around. Just when they come up behind you, you can tell by the smell and the noise. And you get used to not spending money, which, as a Scotsman living in Yorkshire, is brilliant. Anyway, so my solution to this problem was to have 4 krill on 4 different pendulums. Okay, 1, 2, 3, 4, and 3 of the krill would be happy and alive. The other krill had gone to krill heaven and so wasn't swimming. Okay. Now what that means is that 3 of the krill, I did this animation myself. Thank you. Okay, 3 of the krill were swimming and 1 of them wasn't. And all I had to do was subtract the difference between the movement of the krill that were swimming from the movement of the krill that wasn't swimming, because the one that wasn't swimming was being moved just by the fact that the boat was moving around. And the ones that were swimming were moving a little bit further, okay? And we found that this worked. And we got a really interesting result. We got lots of really interesting results, but one of the results in particular was we found that krill suffer from postprandial narcosis, okay? So after eating, sleeping. Okay, Christmas dinner effect. So you have your Christmas dinner, you're full, you sit down, you fall asleep. That happens to krill as well. And that makes for a really interesting carbon pump. So what happens is krill, so we go back to that diurnal vertical migration we talked about at the beginning, the idea that animals move down during the day to hide and come up to the surface to feed at night and then go down. So once a day, they're doing this thing with a pumping carbon. They're capturing carbon at the surface, taking it down. Well, krill come to the surface, feed, sleepy, fall asleep, and fall down through the water column. And they kind of parachute. So the one in the bottom picture there is actually parachuting. It's put all its legs out, and it's just drifting down through the water column. They'll drift down to 40, 50, 60 meters. And then they'll have a poo, and they wake up. And then they start swimming again, and they come up to the surface, and they swim, feed, fall asleep, and drift back down through the water column. And we reckon they do that between 3 and 6 times a day, which turns out to be incredibly important, because krill, there are lots and lots and lots of krill. Somebody did the maths and worked out there are enough krill to cover the whole of Scotland in a layer 1 krill deep. Okay? That's a lot of animals. So they basically have this behavior that we now know results in them. I did this animation myself as well, by the way. We now know results in multiple trips to the surface and back down again. Each one of those trips, if you're a krill, if you're the size of a krill, that's the equivalent of walking up Everest before breakfast and then coming back down again. Abseiling down, I guess, and then walking back up again before lunch, abseiling down again, walking back up. 5.7 kilometers a day is how far a 5, 6, 7 meter krill can travel. That's a lot of swimming. Okay, so how's that relevant to eyes? Well, from my previous work, we've worked out that eyes are tuned to depth. Okay, so animals have eyes that are adjusted to particular depths. If you live at 800 meters is the kind of cutoff point in clear oceanic water. That's the point at which down sunlight ceases to become important. 600 meters, you can still use sunlight to see silhouettes above you, and black stuff below you. 800 meters, it stops being relevant. And so the animals are tuned to those depths. It's not just the tapetum, it's their spectral responses, it's their temperature, how quickly they can respond. All of those things we looked at, and we found that there was a change depending on the depth at which the animals were found. Of course, in the North Sea in particular, we have a problem in that there are lots and lots of nutrients. There's lots of disturbance to the seabed in the North Sea. There's sort of particles coming out of rivers. Look at the Humber. It's brown. It's quite naturally brown, but it's brown. And lots of rivers have been disturbed, so they're putting more stuff into the sea. Chemicals, plastics, rubbish, pollution, all sorts of things coming into the sea. And that's changing the nature of the North Sea. Now, for a lot of years, people have been using this technique. Any of you who have done any sort of field ecology in freshwater, marine at all, will recognize this piece of highly technical piece of equipment. It's called a Secchi disc. The Secchi disc is basically just a circle, black and white. And you lower it down through the water column until you can no longer tell the difference between black and white. So if it's really murky, say you did it in the Humber, you might get 10 centimeters. If you do it in the middle of the ocean, you might get 100 meters. Okay, so it's quite a useful measurement. And because it's such a simple technique, people have been doing it for a long time. And one of my colleagues, Roney Forster, led some research on this, where they looked at this thing called coastal darkening, which is the idea that the seawaters in the North Sea in particular are getting more and more, the Scottish term would be fusty, so they're getting less and less easy to see through. And they measured this using Secchi disc records from history, and you can see that there's this real split between I can't read the date on that. There's a split over times, okay? And you can see that Secchi disc depths have decreased over time in lots of different body waters. This is just a few areas from the North Sea, but you can see that it's changed significantly in spring, summer, autumn, winter, in different parts of the North Sea. So we have this impact called coastal darkening. And so it reflects a change that has happened in the North Sea. So the North Sea used to be full of filter feeders, and it didn't used to get the same inputs of nutrients and debris and stuff. And it used to be clearer. In the Thames, you used to be able to lower a Secchi disc to over 15 meters and still be able to. You'd be lucky to get 5 meters nowadays, just outside the Thames. So that's a significant change and reflects a real change in ecology that has impacts on the animals that live in the sea. If you're a visual predator, lots of animals use their eyes to some degree. If you're a visual predator and your world's suddenly gone fusty, you're going to have trouble or not be quite as efficient as you used to be. You're going to be at the wrong depth as well because you're adapted to a particular light intensity. That's where you want to be. And you're not going to be able to see your food so much. If you're a jellyfish, it's great. Jellyfish don't really care about light. They just swim around and bump into things and feed on them. So there's a change. Right. Menti number 2. This question on here is, it's the same code as last time, but the question on here is, what do you think of when you think of Sperm Point? Those of you, how many of you have been to Sperm Point? Good. Any of you that haven't, talk to that man over there, or these gentlemen over here, and ladies and gentlemen over here, and ask when to come and visit. But I shouldn't say anything because I'm asking you what you think of Sperm Point. Let's just do it old-fashioned way. What do you think of when you think of Spurn Point? Give me a one word. That doesn't work when I'm deaf. I'm deaf. It doesn't help. Bacon sandwiches, that's a good one. Yes? Yeah? Yes? Knots, that's a bird. Yeah? Erosion? Wilderness? Wind, yeah, my God, yes, absolutely. Yeah. Stand in your eyes. So what about military? Military history? Old buildings? Lighthouse? Industrialization? Railway? RNLI station? ABP? Isn't that part of Spurn as well? But it's still a special place, in my view. I mean, I think of wilderness, and I come from proper wilderness up north, you know. But it is a special place. I just want you to bear in mind that Spurn Point is more than just a historical view of nature, okay? So I don't want to tell you what my view is yet, but I have a certain view of these kinds of things. If you go back in time on Spurn, this is an old map from 1936, Phillips. There's a posher version that the Yorkshire Wildlife Trust have put together, but I like the old version. And what the hatched areas show is the extent of seagrass in those areas. And if any of you have been to Spurn, or the areas around, and looked in the sediment, looked in the banks along the beach, you'll occasionally find these masses of oyster shells. And if you look on the beach, there's oyster shells. They're everywhere. They're collections, they're middens of oysters in various parts of the Humber. So in the past, this was a place where there was an environment that had lots of seagrass in it. It has spartina, which is that sort of, I can't remember the common name for spartina, sort of tufty grass. There's tough grass that you find growing slightly above on the salt marshes. And it had oysters. It had lots of oysters. It had so many oysters, in fact, that it was an important fishery. Native oysters in Humber, Cleethorpes mostly, there was lots of them in Cleethorpes. They made ponds so that they could grow them for longer and farm them. You know, it was basically a very simple form of aquaculture. And the Humber was really important. And people that lived in Grimsby and Manchester, instead of going out for a kebab, or vegan kebab, sorry, for those of you in here, instead of going out for a kebab after a few beers, they're going to have some oysters from an oyster bar. So oysters are really an important part of the Humber estuary. When we started the project off, Yorkshire Wildlife Trust, James basically, and I, a long time ago, we were pretty amazed at how interested newspapers were in this thing. So people have a real affinity for this thing that looks like a little grey rock. It's a rock that sits in the water. It's not charming. It's not like a dolphin or a whale or a penguin or anything. It's just a little grey rock, but people feel this real affinity for this animal that they kind of feel should just be there. It's a part of the history and culture of the Humber. So we started this Humber aquaculture project, and it's really small scale at the moment. We've got 7 trestles of oysters. AJ, who's going to talk to you later on about, don't want to talk too much about this because he'll talk much more authoritatively than I can. Basically, we set this up. We were told by everybody that this wasn't going to work. That you can't grow oysters in the Humber because it's too fusty, right? There's too many particles in the water, and the oysters have to sort through the food and the particles they don't want to eat, and it takes up much energy, and there's no way they're going to survive. We were told by scientists in my own institution who are experts in estuarine ecology, like proper up there experts, not like me. We were also told by oyster farmers from Ireland who said you can't grow native oysters on trestles in the intertidal. But guess what? There's a picture. I think, is that your hand, AJ? Yeah. So that's a picture of an oyster, and around the edge you can see that kind of light colour. I think they're growing quite well, aren't they, AJ? Yeah. You're not allowed to say anything else, but you know it's true. They're growing quite well. They're surviving really well. That graph on the right isn't as spectacular as you might want to see, but it's because they're small animals growing by 3 millimeters or something. But they are growing. This is from last year. This is when we didn't really know what we were doing before we got AJ involved, and we started thinking about, well, what's the best place? Well, we were still working out what the best place to put the oysters was in terms of tidal height and all the rest of it. So oysters grow in the Humber. They can grow in the Humber. We can bring them back. And we have the remnants of that ecosystem that was there in 1926. It's still there. We've still got seagrass. We've still got spartina. Now we've got native oysters again. There may even be native oysters lurking somewhere in the depths. We do have, not native oysters, but the American oyster, which was imported into the Humber. Caused a few problems, but it was imported and it's still there. It's still growing on the pier legs across in Grimsby. But there's the makings of a rewilded Humber. It's not starting from zero, we're starting from quite a low base, but there's real possibilities there to do something. When we put the oyster bags in place in the Humber, biodiversity increased immediately. I think that was a week after, about a week after. Is that your photograph? Yeah, so James was wandering around, and there's a seal sitting on top of an oyster bag that's full of oysters. So we increased biodiversity. What we want eventually is to see something like this. Basically, the temperate equivalent of a coral reef. Okay? Just as complex, nearly as diverse. Lots of interesting things going on there. Lots of linkages. High levels of resilience. Okay? We want this to be a rewilding that survives the changes that are definitely coming, right? So we've seen changes in our lifetime. We're measuring changes in the last 10 years, 20 years. Change is going to keep going and probably going to accelerate. We need to be doing stuff that is resilient to those changes. Why are they such a good idea? Well, here's, these reefs, these oyster reefs provide shelter for other organisms. They increase biodiversity in that way. They help with coastal protection. They encourage other organisms to settle locally because oysters take stuff out of the water, make it a little bit clearer. And if it's a little bit clearer, you've got a better chance of surviving if you're a piece of seagrass or another bit of algae under the water. They encourage worms and stuff which suck carbon from the surface and bury it at depth. So they're part of the carbon cycle. The oysters incorporate carbon into their shells, part of the calcium carbonate matrix. Then they make these sort of dead shells that lie around, which encourage other things to settle. They add to the complexity of the environment. So as you add complexity to environments, you make more niche space, more space in which other animals can come and settle. So these are a really good idea, and it's much better if we can do it using natural processes rather than trying to use artificial processes. So what can we turn things back? Can we go back the way? Can we go in reverse and go from an environment which is cloudy, fusty, low diversity, dominated by sort of scavengers rather than pelagic feeders, low levels of algae. The levels of phytoplankton off the coast here, where it's quite fusty, I keep using that word fusty, so quite dark, are lower than anywhere else around the coast of the UK. It's surprising we've got such a good lobster fishery. Something else to talk about later on. But anyway, moving on, because I need to. These are also found in the Humber. These are all invasive species. So we've got the Chinese mitten crab, zebra mussel, there's an amphipod shrimp, I can't remember the name of. Dicherogammarus, that's it, zebra mussel, Crassostrea gigas. It's not called Crassostrea anymore, but that's the American oyster. That's also an invasive species. Okay, they're all there. They're here already. Okay, they're not going to go anywhere. We're not going to get rid of any of these things, unfortunately, because they're established. So don't bother with the Menti question. It's not working. It's not working for me. So my last question, and it's a kind of yes or no answer, really. We want to re-establish the ecosystem in the Humber. I want to see sturgeon back in the Humber. I can see James shaking his head no. I want to see sturgeon back in the Humber, right? We have native oyster, and they are managing to grow at the moment, but they are less resilient than the American oyster, which is here already. How do you feel about using a species for rewilding that does all the jobs that the native species use? This isn't an easy question, or there's no real easy answer. How do you feel about using an American oyster, which builds reefs better than the UK, European oyster, to re-establish that ecological function. So you've got a choice. You can vote for either Ostrea edulis, which is a native oyster, which is what should be here, which is what we've been working with until, what we're working with to try and re-establish oyster reefs in the Humber at the moment, but is unlikely to survive the climate change that's coming around the corner. I mean, we're talking 2-3 degrees in the next 20 years or whatever. No, not 3. I'm just trying to scare you. Okay, the next 50 years. Or can we use Crassostrea? Right? So I'll let you vote. So all those in favour of Ostrea edulis, raise your hands. One. Ostrea edulis, that's a native oyster. All those in favour of native oyster. I think that's just probably around just over 50%. But those in favour of the Crassostrea, just to get a... If there's more than 50%. Yeah. Who doesn't care? So I don't know what the answer is. Personally, I like the idea of using something that works, and probably a mix of the two. Yeah, okay. So a mix of Ostrea edulis and Crassostrea, guys. A mix of the native and the American. They may help each other. Other places have not found that to be so, but every case is different. Okay. Other places have found that oysters aren't helpful in carbon capture, but we think they are here. So every place is slightly different. So, thank you for listening.